100 years ago, Victor Hugo wrote, "Where the telescope ends, the microscope begins. But which of the two has the grander view?" Today, we still seek the answer. A Fine Science with Holly Preston.
Plant biotechnology, in the strict sense, is the ability to go into cells, the building blocks of all living things, and change at will the structure and functioning of those cells. That is the definition given to us by Dr. Warren Steck of the Plant Biotechnology Institute in Saskatoon. The Institute does research in many areas, but the bulk of the work is geared to agriculture. The general purpose is to make plants that will better serve our needs. On this program, we'll look at three of those areas. One called molecular genetics, one that is studying the chemistry of the flowering of plants, and another concerning nitrogen-fixing plants. Also on this program, three research projects in the Crop Science Department of the University of Saskatchewan. The first is also in the plant biotechnology area. It's an attempt to develop plants that will grow in saline soil. The second is studying why plants freeze, and the third evaluates the quality of agricultural crops. But first, the Plant Biotechnology Institute. Some of the areas in this division of the National Research Council are involved in very basic and exacting sciences.
I'm involved with a section on molecular genetics, and molecular genetics within the biotechnology realm really has as its mandate trying to understand in precise molecular terms how cells chemically express their genetic information. And that's a very basic sort of a science endeavor. In fact, it's still very junior days for this kind of science, since it really didn't evolve to the mid-70s. But with some of the basic things that are in hand, this understanding of the way that genetic information is expressed is a prerequisite to being able to take genetic information and exchange it between organisms that otherwise don't have it. For example, cloning pharmaceutical peptides into bacteria and getting them to make insulin, this sort of thing, all stands on a basic science prerequisite of understanding how genes express their information. And really this section has as its mandate an understanding in molecular genetic terms of how plants do that. How do plant cells take their genetic information and express it? The ultimate objective, I think, is really twofold, and my lab and the other couple of labs in this section are concerned in this way. One is to continue to work in the basic aspects of trying to find out the rules by which plants express their genetic information. That's one. And then taking that information, going off in a not entirely separate but a second direction, and that is taking those rules and using them to exchange genetic information either between plants or taking genetic information from animals, from bacteria, yeast, whatever, and seeing that they are expressed in plants.
Other scientists have very specific research projects, such as studying the chemistry of the flowering of plants.
Nobody has discovered exactly what makes plants flower. How do they tell out in the world that it's time for them to turn from a vegetative plant into a flowering plant? And there's been lots of evidence, and it's been a big mystery since about the 1930s. People have known that there was something that makes plants flower. The plant we're working on is not this one here, but we can demonstrate it. The plant we're working on is called the cocklebur, and it grows down on the banks of the Saskatchewan River around here, and it's got little burrs on it. And it is known, and has been known for a long time, that when the summer comes to an end and the nights get longer, that the plant decides all of a sudden it's time to flower. And they know that the trigger that makes the plant turn from a vegetative plant to a flowering one is the photoperiod, the length of the night. And we can take these plants from the riverbank and grow them in the greenhouse and fool the plants into thinking that fall is coming and you can make these plants flower. And you can take a plant that's been fooled into thinking it's time to flower and cut a leaf off of it at the stem here and graft it onto a plant that has been sitting under summer conditions, and that plant will flower. So we know that there is some signal in the leaf that is being transmitted from that leaf into the receiving plant. There is all kinds of evidence to say that there's some substance that's being sent from the leaf up to the top of the plant. And what we're doing is we're looking for that particular substance in collaboration with some people in Ottawa who are doing the biology. Now, a scientist can't be everything at once. You try to do as much as you can, but our object here is to work on the chemistry of the problem, to try to isolate this substance and to figure out what its chemical structure is.
What would be some of the practical applications of a success?
Well, this is really a glamorous project in terms of its possible applications. Particularly here on the prairies, if we could isolate and make a substance that would induce plants to flower more early, we could possibly extend the growing season of plants. And that would be really useful if you could get a couple of more days or maybe even a week or two of extra growing season in, then you could grow plants here better and different plants than you could grow otherwise. Also, you could get a crop to flower or to mature all at once so that you could simplify harvesting really a great deal. Also, you could potentially kill off a crop that you didn't want, like a weed at the roadside, by having it flower too early so that it wouldn't set seed properly, and you could, in an environmentally safe manner, get rid of a plant that you didn't want without having to use herbicides that were harmful.
How are you progressing? How's it coming along?
Well, it's a very difficult problem because only a very small amount of these substances are produced, and this is why the answer to this problem has eluded people for a long time. The quantity of the magic flowering hormone is going to be so very, very small that it is difficult to isolate it. And I'd say we're doing very well. We have, with our collaborators in Ottawa, a biological system for telling where the flowering substance is. We can isolate it, get it into test tubes, put it back into plants, and make those plants flower. So we have a way of telling how we're doing in our separation of the chemicals. We have a long way to go, but I think we're making pretty good progress.
Nitrogen fertilizers are an intricate part of high-yield farming today. They're also very expensive. Dr. Louise Nelson is working on another possible source of this essential element.
There are certain bacteria which are found commonly in the soil which have the capability of forming nodules on legume roots. This is an example here of a nodule. Legumes are things like peas, faba beans, lentils, clover, alfalfa. And when these bacteria are in the nodules, in the roots, they're able to fix nitrogen. That is, they can take nitrogen from the air and turn it into ammonia, so they act as a natural fertilizer. And this, of course, is of interest because the cost of commercially produced fertilizer is growing. There's always a shortage. Most plants require nitrogen, and so these organisms are able to provide this nitrogen. It's not a completely free process in that it requires some energy from being provided by the plant in order to support fixation. The problem is that there often aren't the right bacteria in the soil, or that they're not the most effective, and so we're interested in finding the right combinations of plant and bacteria which will give the most nitrogen fixation and produce the best plant. We're looking at a number of things. One of the problems we often find in agricultural soils in this area is that there is too much nitrogen already in the soil. This may be as a result of naturally occurring soil nitrogen, or because fertilizer was used in the previous crop and there's still some left in the soil. And when there's sufficient nitrogen present in the soil, the process of nodulation is inhibited, and we're looking to find strains of bacteria and plants which will continue to fix nitrogen even in the presence of low levels of nitrogen already in the soil. So this is one of the main things that we're looking at. It might be of interest, for instance, in crop rotation. You might want to, instead of using summer fallow, you might want to rotate legumes in combination with the grain that you happen to be growing. This is being promoted more and more in this province. In the northern part of the province, just north of Saskatoon, legumes are grown commercially. Peas, faba beans, lentils are now being produced in quite substantial acreage as potential food source for animal or human consumption. Another long-term aim is to transfer the capability of these bacteria to fix nitrogen to non-legume systems, and this may involve either finding bacteria which are able to nodulate things like wheat or rapeseed, or else actually transferring the genetic information for fixation into the plant gene. But this is a very long-term process, but it's something that we're thinking of as an eventual goal. I should mention that I'm working as part of a group. I'm interested primarily in the bacterial side of things, but we also have members of the group who are interested in plant physiology and plant genetics. And one of the things that perhaps we should be looking at is developing plants which are drought tolerant, because this is a major problem. Unless you have irrigation in the southern parts, the legumes don't always do that well.
From Quality Records, a musical evolution, the ultimate fusion, the majesty of symphonic splendor, and the driving power of rock and roll. The Vienna Symphonic Orchestra plays today's hottest hits as you've never heard them before. This is VSOP. A new generation of musical entertainment. Today's biggest hits, bigger than you've ever heard them before. You'll get St. Elmo's Spire, Rock Me Amadeus, Kyrie, A View to a Kill, and Private Dancer. The classic sound of rock and roll. Welcome to the Pleasure Dome, the power of love, and Hearts on Fire. VSOP, the Vienna Symphonic Orchestra Project, on LP, cassette tape, or compact disc, is in stores now.
Not too far from the Plant Biotechnology Institute, similar research is being carried out at the university. The Crop Sciences Department is working on a project to return thousands of hectares of agricultural land to productivity. In many parts of Saskatchewan, crops cannot be grown because of the salt concentrations in the soil. Efforts to recover the land itself have not been very successful so far. But Dr. Alan McEwen is looking at another approach.
We're developing salt-tolerant crops for Western Canada because approximately 10 million acres of our land are affected by salts to the point where crop yields are reduced. So it's a major problem. It's something that plant breeders don't really have a handle on because there are not a lot of conventional mechanisms for salt tolerance that they can use in their breeding programs. We're trying a new approach called somoclonal variation, which is a type of biotechnology. The way it works is that we know that in each plant, composed of millions of cells, each cell has the entire genetic capability of producing an entire plant again. But a root plant or a root cell, for example, would have all of the genes normally turned on in a leaf. They would be turned off in the root, and vice versa, of course. What we do is we take the cells out of the plant and we stimulate those cells in such a way that they grow prolifically. They grow into a large ball of what we call a callus, which is an undifferentiated or unspecialized group of cells. And when they undergo this rapid growth, proliferation, they also undergo genetic changes. And we think that what happens is that somewhere in the genetic capability of these cells is a mechanism for salt tolerance that has been turned off generations ago. And this proliferation, or the callus growth phase, is a way of turning those genes on. We grow millions upon millions of these cells in a small petri dish, and then in order to decide which of these millions of cells has undergone the change that would perhaps permit the cell to be salt tolerant. We transfer all of the cells into a culture medium containing the nutrients and hormones it needs, plus an excess concentration of salts that we figured would normally be lethal to the crop plant cells. We identify the ones that survive. Typically, upon transfer, the cells would die in this medium, but occasionally we see some cells that remain green and healthy when all the neighboring cells die. We presume that some change has gone on in these cells to cause them to be salt tolerant. So we then pick out those cells and we put them into a new culture medium, this time containing the proper nutrients and hormones to cause a reorganization of the cells into shoots and roots. And ultimately, we grow these out into whole plants in the greenhouse. Then we take the seeds when these plants mature and we grow them in both normal soil and saline soil to test whether the plants actually are salt tolerant or not.
Are you finding some?
We're finding some with flax. Flax is the system that we've developed furthest so far. We have over 40 plants that are presumably salt tolerant. 2 of these plants we've tested in second and later generations in saline soil, and one of those 2 does appear to be salt tolerant as well as high yielding under normal soil conditions. So we have hopes for that.
So what's this going to mean if you actually can develop a plant that's going to be commercially viable that will grow in a very saline environment?
Well, as I mentioned, about 10 million acres in the Canadian prairies are affected by salts to the point where crop yields are reduced. Of course, we're trying to recover some of that lost yield by developing not only salt-tolerant flax, but we're also working with wheat, our most important crop. Unfortunately, wheat doesn't respond to our techniques the same way flax does, and it's a bit more difficult to go through these rather routine techniques, or at least routine for flax. So far, we have developed one wheat plant that is presumably salt tolerant, but we haven't had the opportunity to test it in saline soil yet.
Any of us who have gardens know the inconvenience of covering plants and vegetables in the spring and fall to try to keep them from freezing. Even worse is an unexpected frost that devastates the plants. So far, farmers can't protect their fields from frost, and losses to freezing can be disastrous. Research is trying to solve the problem.
The primary objective of this lab is to study how plants survive our winters and how they tolerate frost. Western Canada, we can get a frost any month of the year. It's not uncommon to have frost 12 months of the year here in Western Canada. And freezing and frost is a very important part for crop production in Canada. We lost crops in Western Canada in 1979. On August 13th, we had a frost came through, wiped out the crop. We lost a crop in 1982. It was a result of millions and millions of dollars of loss to the farmers. From research in the United States, mainly out of California and Wisconsin, that if there's certain bacteria on plants, growing on the plant, it will cause the plant to freeze right around -3°C. Now these bacteria are not harmful to the plant, they just exist on the leaves, but if the temperature does drop below 0 degrees, it causes the water to freeze in these plants. Water itself does not freeze at 0°C, contrary to what many people think. Water actually, you can cool water down to -40 degrees, it'll freeze then by itself. But a lot of our water does freeze right around -5, 6 degrees due to impurities in the water or ice crystals floating around in the air. This bath here is right now at -4°C, and theoretically the water should be frozen. In this test tube I have ordinary water. You see it's sitting at -4, it's not frozen here. Now this water, we call it supercooled water. It remains in an unfrozen state, even though at 0°C it'll freeze. And for example, if I added an ice crystal to this test tube, it should theoretically freeze here. See the change in color of the water? The water is freezing. The water has potential to freeze, but it remains in a supercooled state because it has no nucleators present to cause it to freeze. Now this water here is frozen, it's converted to ice. It's when you have ice in plants, this is what causes damage to the plant. Here's an example where we've added bacteria to ordinary water. This is not all bacteria cause freezing. There's only certain bacteria that do it. In this case, is Pseudomonas syringae. And this will cause water to freeze right around -3 degrees. And this is a very important bacteria in the aspect of causing freezing injury in plants in Western Canada, or in that matter of fact, all over the world. If these bacteria were not present on leaves of plants, the plants will not freeze. Here's an example where a leaf has been treated with these bacteria and cooled to -3 or -4 degrees Celsius, and you see it's frozen. When the leaf freezes, it causes the water to freeze around it. And we use this as a test for the presence of these bacteria. And this leaf here now is frozen. It'll die, it will be thawed out. Here's an example where the leaf has no bacteria on it, no nucleating bacteria, and it's sitting here at -4 degrees Celsius and it's not frozen. And if you take this leaf and warm it back to 0 degrees Celsius, it'll be perfectly healthy, and it actually escapes the freezing injury here. And this is an important aspect of our research, finding ways of controlling these bacteria and see how they change during the growing season.
Winner of every major instrumentalist award, Roy Clark now plays some of the greatest songs of all time. These beautiful recordings are sure to stir beautiful memories. This treasured collection of all-time favorites played by Roy Clark. Plus, you also get a second album including Roy Clark's greatest hits.
Yesterday, when I was young, so many
These million-selling records are loved throughout the world.
Come live with me and be my love.
And this second album contains still more great instrumentals with the Beatles' Yesterday and Norwegian Wood. Love Me Tender, the classic Malagueña, and the immortal San Antonio Rose. Remember, you get both treasured albums of all-time favorites played by Roy Clark, plus Roy Clark's greatest hits on 2 records or 2 cassettes. Available only through this TV offer, so order now.
Visa, MasterCard, COD charges. Call 569-1321 or save COD charges by sending $14.98 for 2 LPs or 2 cassettes, plus $2 postage and handling to Roy Clark, P.O. Box 124, Regina, Saskatchewan. That's Roy Clark, P.O. Box 124, Regina, Saskatchewan. Please specify records or cassettes.
Modern technology has already produced many high-yield, high-quality agricultural plants. At the same time, scientists are constantly trying to improve on those plants and breed new ones. There's a department that evaluates their performance.
This laboratory is called the Crop Quality Laboratory of the Department of Crop Science and Plant Ecology. What the crop quality does is to analyze those constituents of grain which we grow in this province for certain constituents which help to sell that particular crop. And at the same time, this laboratory does research on certain other constituents of those grain which have harmful or deleterious effects on crop quality. Let's take, for example, of cereals. In wheat, if we are talking about bread wheat, we need a wheat variety which has the highest bread making quality. So we would analyze those samples or those lines of wheat plants for protein, for flour yield, for baking qualities, things of that sort. Similarly, for barley, which goes into, let's say, feed area, we would analyze them for high protein content, and which of course the farmer needs to feed his cattle. And similarly, we'll analyze for high energy, because after all, cereals in animal feed provide energy. Okay, same way in the case of oilseed or flax, which we are talking about here, there are generally two broad areas into flax oil goes. One is the industrial oil for the production of paints. Now, and the second type of program in flax production, which we are recently looking here, is to produce flax which has an edible type of oil. Okay, same way in legumes. Like, for example, in lentils, all the lentils go into the soup trade. So we determine the cooking quality of lentil, because that is very important. In other words, lentils should cook in a very short period of time, and they should provide a certain amount of consistency when they're cooked. So that is what we call cooking quality. From about September till March, this laboratory does all those quality criteria in the samples which are brought in into the lab. And then in March, all this information is given back to the plant breeders, who decide then which line or which particular line of crop he has to grow for the next year. Now, in terms of dollars, this, of course, agriculture being the largest industry in our province here, even a small increase in yield or a small increase in quality or improvement in the crop quality really can translate into very large sums of money. So that's why we think our effort here, along with of other people in this department, is to serve the farmers of Saskatchewan in as best way as possible as we can.
And that's our program. We hope you've enjoyed our look at some of the plant research going on in Saskatchewan. From what we've seen, the province seems to be a hotbed of research in the area of agricultural crops. It seems fitting in a province that depends so heavily on farming. I'm Holly Preston. Thank you for watching A Fine Science.
This is CBC Television.